study different a bit different way. So what is the basis of classification of the Protestant? So primarily Protista. So put the heading classification of Protista. Classification of Protista. So we have already studied general characteristic of Protestants. So let's start with classification of Protista. So when we study the classification of the Protista, basically the Protestants have been classified on the basis of nutrition. So on nutritive basis, we have classified Protestant in 3 categories, right? So first, those Protestants, so they are based on, so this classification is based on nutrition. So if we'll talk about the nutrition, so classification of the Protista, the first Protestants are those which are photosynthetic Protestants. Photosynthetic Protestant. So photosynthetic Protestants are those which are basically which are green form, or they have photosynthetic ability and they make their own food. Means basically they all are nutritionally, they all are autotrophic. Second category is that is they are consumer decomposer. Consumer decomposer. So consumer decomposer they are something a mid value of we can say fungi and like animals. So they are basically decomposer, and they belongs to, they have similarity with the fungi. So they grow or they trade nutrition from dead and decaying material. But sometime when there is scarcity of food or scarcity of dead and decaying material, what they do, they start behaving like amoeba like organism, and they start phagocytosis, and by this way they becomes consumer. So that's how we have classified them on the basis of their nutrition. And the third one, they are protozoan Protestant. Protozoan Protestant. Basically, they are animal cells. Protozoan Protestant. Now again, we have further sub-classified these photosynthetic, consumer decomposer, and protozoans. So you will find number of the heading in your MCRT, but we are studying in a proper systematic way. So systematically, we have classified these Protestant in these 3 categories. Further, the photosynthetic, under the photosynthetic Protestants, we are studying like Chrysophytes. So first one are Chrysophytes. So Dinoflagellate, then Chrysophytes, and then it comes to the like the third one. The third one, they are euglenoid. We'll study in detail what are the Chrysophytes, what are the Dinoflagellates, but there is a similarity between all these 3, that they all make food with the help of photosynthesis process. Means they are autotroph and they have capability to make food by the process of photosynthesis. Next, this is consumer decomposer. So when we talk about the consumer decomposer, there is only single class of or single category of organism. They are called slime mold. So we study slime mold. Again, protozoan Protestant. In protozoan Protestant, when we talk about the protozoan Protestants, so they are basically animal cells. So they have resemblance with the animal and their characteristic matches with the animal. So further, these protozoans have been divided. So the first, on the basis, we further divide protozoa on the basis of locomotion. So we divide on the basis of locomotion. So on the basis of locomotion, first protozoa, they are called flagellated protozoa. Actually, these flagellated protozoa are those which have flagella for locomotion. The second one are amoeboid protozoa. Amoeboid protozoa are those protozoa which have locomotion like amoeba, means they locomote with the help of pseudopodia. Then it comes the sporozoan. Sporozoan protozoa. They are those protozoa which do not have any locomotory organ, specific any locomotory organ. So the fourth one or the last one, these protozoa, they are called the last protozoa. They are ciliated protozoa. So ciliated protozoa like Paramecium. So this is the actual classification of Protista. Clear? Please note it down and then let me know. Then we'll start with photosynthetic protozoa from Dinoflagellates only. Note it down and tell me after noting down. Done. Okay. So fine. So put the heading photosynthetic protistans. Photosynthetic protestant. These organisms constitute, note it down, in photosynthetic protistans. These organisms constitute main part of phytoplankton. Main part of phytoplankton. What are phytoplankton, by the way? Phytoplankton, those small unicellular photosynthetic form which are taken as a food by fish and crustacean. Note it down. Phytoplankton are free floating green photosynthetic organism. They keep on floating in water. And are the major part of food by major part of food for small fishes and crustaceans. Now the first photosynthetic, yes.
Sir, एक बार please repeat कर दीजिए from starting phytoplankton.
Okay, fine. So note it down. Phytoplankton are the green photosynthetic organism. Which are photosynthetic organism which keep on floating with water stream.
Sir, this is Vaishnav, sir. Sorry for the delay, sir.
Okay, no issue. So Vaishnav, like we have started with in last class we have summed up with the general characteristic of Protestants. So you can take the screenshot of this one. After the general characteristic of Protestant, we have discussed this type of the Protista. Okay, so let me complete the sentence, then I will explain it again. Just give me one second. So yes,
Okay, sir.
yeah, note it down. So they are the major part of food. For fishes and crustaceans. Clear? Okay. So I have told them that we divide Protista into 3 categories on the basis of nutrition. So first category is photosynthetic Protestant, second category is consumer decomposer Protestant, and third category is protozoan Protestant. So in first category, that is photosynthetic Protestant, they are those Protista, means eukaryotic organism, which make their own food, means they are autotroph, right? So further we divide photosynthetic Protestant in further three subcategory. First Dinoflagellate, then Chrysophytes, then euglenoid. Again, in the second category of Protestant, they are consumer decomposer, and under this there is a single subcategory, we call them slime mold. The third type protozoans are, they are Protozoa Protestants. So Protozoa Protestants, they are four types. They have four subcategories. So we have again divided these Protozoa in four categories on the basis of locomotion. First of them are Flagellated Protozoa. They move with the help of flagella. Second are Amivoid Protozoa, which looks like amoeba. And third is Sporozoans. They do not have any locomotory organ. And the fourth one are Ciliated Protozoa. They have cilia for locomotion. So this is the classification of Protozoa. Clear? Take the screenshot. Vaishnav. And then again, we are going to start with photosynthetic Protestant. So in photosynthetic Protestant, so further photosynthetic Protestant are, in general way, they are called phytoplankton. So they are small unicellular green forms. They keep on floating on the water, and actually they are used by small fishes and crustaceans for sake of their food, right? So small fishes, small vegetarian fishes, and small crustaceans, they keep on feeding on these phytoplankton, and they are basically what? They are basically photosynthetic form of Protestants, right? So the first subcategory is Dinoflagellate. So what are the Dinoflagellate and what are the characteristic of Dinoflagellate? So A is Dinoflagellate. Dinoflagellate. So let me explain you first, then we will jot it down. What are the Dinoflagellate? Actually, Dinoflagellates, they are the photosynthetic form, and they have peculiar sculptured body. So I'll show you the diagram of Dinoflagellates. An example of Dinoflagellates are, there are two examples. So example are Goniolex and Gymnodium. Gymnodinum, just a minute. Gymnodium. So we pronunciate them Gymnodium, and we write it down Gymnodinium. And the next are Goniolex. So Gymnodium and Goniolex, they are a type of this one, Dinoflagellate. So let me explain you what are the Dinoflagellate. Let me make you more clear. Just I'm going to show you a diagram of these two. Protista. Dinoflagellate. Look, this is the diagram of Gymnodium and Dinoflagellate, right? So this is the diagram of Dinoflagellate. So you will find this is Gymnodium and this is Goniolex, right? And they are also the rest two one. They are also Dinoflagellate. Look, so their body have certain design or pattern, like they have sculpture, right? So they have plate-like structure. That's why we also call them armored Dinoflagellate. Generally, these Dinoflagellate, they have two flagella. So the first flagella is transverse flagellum, and the second flagella is longitudinal flagella. So generally, they have two flagella, but both of the flagella, they originate in different direction, right? And their covered is made up of cellulose plate. So we call these covered theca or lorica. So they have these cover-like structure, and this cover is made up of cellulose. This is called theca and lorica. Actually, they are photosynthetic form. They keep on floating. They perform photosynthesis. Some of them illuminate the light. Hence, they create the phenomena of bioluminescence. We call that phenomena bioluminescence. What happens during the night time? They illuminate the light. That's why they glitter in the water. So we call this phenomena bioluminescence. So this is the question of medical examination, which has been asked many times, that which of the following, many time, number of the time it has been asked that which of the following organism is responsible for bioluminescence. And normally the answer of bioluminescence is that is this one, Goniolex. So Goniolex is the dinoflagellate which illuminate light in night, and it perform the phenomena of bioluminescence. For more important question which has been asked in your medical examination, in like in the highest medical examination, that is AIMS, and even in NEET examination. So the question was, which of the following is responsible for red tide? What happens? There are some Gymnodium and Goniolex. These Gymnodium and Goniolex, these two, they have red color pigment in their body, and when they flourish or multiply in large number, the water become of red color. And whenever the water, the tide comes, it look like a red tide. And these two are responsible for red tide phenomena. So Goniolex and Gymnodium are also responsible for formation of red tide. So generally in medical examination, you will get question which of the following is responsible for red tide phenomena, and the answer will be either Gymnodium, Goniolex, or both will be the answer, because both are responsible for this phenomena. Is it clear? Clear?
Yes, sir. Yes, sir.
Crystal clear. Fine. So take the screenshot of Gymnodium and Goniolex and please draw it. Fine. So let me tell you some more important. Look, I don't know I have told it or not. So look, this is in general a concept of biology. Suppose that there is an organism. Suppose that there is an organism and it have two flagella. Flagella originate from this type of structure, and these are called blepharoplast. And if both flagella are similar flagella, so this kind of flagella, this kind of flagella are called Whipless Flagella. Whiplash, right? So these are like whip tail like flagella, like means hunter like flagella. So these are structurally similar and direction is similar. So when two flagella are similar, so look, if flagella are similar in structure and position. If two flagella, they are similar in structure and position, this kind of flagella are called isoconed flagella. Isoconed flagella. So if two flagella, they are similar in structure and direction, or we can say position, they are called isoconed. Suppose that if they are similar in structure but different in position, one flagella is here and one flagella is here, so direction is different, so they are similar in structure but different in length or position. We call them the second kind of flagella, they are called heteropond. Heteropond flagella. So heteropond flagella are those. There is one more type of the heteropond flagella. Suppose that there is an organism. It have flagella in same direction, means same position, anterior flagella. But the thing is that this flagella is smooth and this flagella is not smooth. It have this type of hair-like structure, further sub-hair-like structure. So these are flagella, they are similar in position but different in structure. This kind of the flagella, this is called whiplash. This is whiplash and this is tinsel. So they have two flagella which are similar in position but different in their structure. So they will be also called heteropond. So heteropond flagella are those which are either they are similar in their different in their structure, or they are different in their position, or they are different in their length. So there are two kind of the flagella, isocont and heteropond. Clear?
Yes, sir.
So please note it down and let me know which kind of the flagellate these dinoflagellate have. Which kind of the flagella the dinoflagellates have? Hey, they have the flagella which is different in direction. So what is that?
Sir, Heterophone Flagella.
They have, be
Sir,
louder.
Heteropond.
Yeah, they have heteropond flagella. Vaishnav, are you getting my point? Did you got it or not?
Sir. Yes.
Okay, great. Note it down. So
Yes,
I'll keep on saying that, suppose that whenever we will discuss in algae, even I will say they have two flagella. One is a tinsel type, another is Whipless type. So you have to understand that this is a heteropond type of the flagella. Red algae, in red algae, there are some gametes which have heteropond flagella. Note it down.
Yes, sir.
Great. Okay. So I told you that these dinoflagellates have different structure. So we were discussing that suppose that this is a dinoflagellate. This is a dinoflagellate with look. So they have two flagella. So one flagella is like transverse and another flagella is like vertical. Now I want to tell you that they have some plate-like structure, sculpture-like structure. So they have armor-like structure. They look like armor of a soldier. And these plates are made up of, these plates are called theca or lorica. Theca or lorica. Theca or lorica, these armor or shield are called theca or lorica. These theca or lorica, they are made up of cellulose. They are made up of cellulose. So these are made up of cellulose, right? And they have heterocone flagella. Heterocone flagella. They have heterocone flagella, right? Clear?
yes, sir.
Now please start noting down. First point. Most of them are marine. Most of them are marine, but some of them, most of them are marine, but some of them occur in fresh water. But some of them occur in fresh water. Next point. So I told you two example, Gymnodium and Gonalax. So note it down. Gymnodium, Gymnodium and Gonalax. Gymnodium and Gonalax grow in large in number, grow large in number. Gymnodium and Gonalax grow large in number in sea, and make water look red, and make water look red. Comma. The phenomena is called red tide. The phenomena is called red tide. Full stop. Next point. Goniolex produces or emit. Goniolex produces or emit light. Goniolex produces or emit light, and this phenomena is called bioluminescence. This phenomena is called bioluminescence. Just a minute. This phenomena is called bioluminescence. So that is emission of light, biologically emission of light.
Sir,
Yes.
sir, only Goniolex emit light.
No. There are two, Goniolex and one more example. If you want to note it down,
Okay.
you can. Noctolica.
Okay, sir, both of them.
Yeah, Noctolica. Let me. But this is the question actually. It has been asked 2 or 3 times. Goniolex. So Noctolica, not Gymnodium. Gymnodium is not emitting light, right? Gymnodium and Goniolex, they both are responsible for red tide. This one. Let me make more clear. These are responsible for red tide phenomena, and then again the bioluminescence phenomena, Goniolex, and one more, that is Noctolica. So Goniolex and Noctolica are responsible for bioluminescence. Goniolex and Gymnodium are responsible for red tide. Clear?
Yes, sir.
And there are so many number of the forms which are responsible for it, but Goniolex is very important. Keep it in mind. So always remember 2 things: Goniolex and Gymnodium. Next point. Cells are generally covered with rigid. Cells are generally covered with rigid structure called theca or lorica. Cell is generally covered with rigid structure called theca or lorica. Full stop. It is made up of cellulose. It is made up of cellulose. Full stop. Because of this covering, they are also called armored. Because of this covering, they are also called armored dinoflagellate. They are also called armored dinoflagellate. Full stop. Next point.
Sir, they are also called
Armored dinoflagellate. I have written it. Armored dinoflagellate. Next point. They have chlorophyll A and C. They have chlorophyll A and C as they are photosynthetic. They have chlorophyll A and C as their photosynthetic pigment. Next point. They have starch and oil as reserve food. They have starch and oil as reserve food. Next point. Asexual reproduction takes place through asexual reproduction, takes place through cell division. Asexual reproduction takes place through cell division. So this was all about the dinoflagellate. Clear? Clear to all of you?
Yes, sir.
Okay, great. Vaishnav, clear? So we were discussing dinoflagellate. Now we are going to discuss chrysophytes. So put the second subheading. The second subheading is chrysophytes.
Sir, did you call me?
Yes.
Sir, I cannot hear you, sir. I have some mic problem.
You have some like
Mic problem, sir.
Okay.
No, sir, mic problem. It's not on.
It's not audible to you.
No, no, sir. The
Okay,
mic
your
is-
mic. Okay, you have some mic problem, so you are not audible to me. Okay, fine. I got it. Fine. So clear. So you can do one thing. You can communicate with me with the help of message, right? No issue. So next, there are chrysophytes. So let's start with chrysophytes. So the example of chrysophytes, they are diatoms or desmate. Datum or desmate. So first, let me make you clear and let me make you understand, like what are the diatoms. Give me just 1 second. Hold on. Okay, so look, diatoms and chrysophytes, in case of diatom and chrysophytes, they are found actually in shallow oceanic water, right? They are found in shallow oceanic water. So what happens? These diatoms keep on floating on this surface. Actually, what is the peculiarity of these diatoms? This is the actually structure of diatom. In fact, they look like this one. Okay. So around the diatom, deposition of silica take place. So what happened? The silica deposited around them. So due to impregnation or deposition of silica, their outer surface become very tough, very rigid. So this is silica. So their outer cell wall is made up of deposition of silica. And after the death of diatom, this silica or their shell do not degrade easily, right? In fact, do not degrade for hundreds of years. It remains intact in this form only. So after the death of diatom, what happened? They keep on depositing on this surface. So they keep on depositing on this surface, and they make a layer, or in fact a thick layer of soil or thick layer of their dead body on the surface of, or we can say on the floor of that ocean or lake where they are found. So this structure, this they are pile up this soil which has been made by the dead body of diatom. This is called diatomoid or diatomaceous earth. This is very important. This is called diatomoid, diatomite or diatomaceous earth. Diatomaceous earth. This is diatomite or diatomaceous earth. So what people do, it is very important economically. So this diatomite is extracted from these oceanic basins or lake basin, and they are used for making filter, for filtering antibiotic, for filtering oil, for filtering sugar syrup, and for filtering various kind of the filters are made by these diatomites. In fact, they are used in our toothpaste as well. So their shell is used in making toothpaste as well, right? So this is the use of these diatomite. And these are the major part of phytoplankton. Like when we were talking at the starting of the chapter, I talked with you that what are the phytoplankton. They are the major source of food for fishes and crustaceans. So actually, out of all these protistants, these diatoms are the major source of food for fishes. Clear? Is it clear?
Yes, sir.
Okay. Now I'll make you to jot it down all these points, but before dictating this point, I would like to show you more and more diagram. Now let me show you the diagram, actual diagram of diatom. Diatoms. Actually, diatoms are their body structure is like this small tiffin box which kids use when they go to their school. So you provide them a small tiffin box, so they look like this tiffin box. So the outer lid of tiffin box is called epitheca. Look, epitheca, and lower lid of lower part of base of tiffin box is called hypotheca. In the side wall of this tiffin, like this is the side wall of tiffin, so outer lid is called epitheca and lower is called hypotheca, and this side wall it have certain design. These design elements are called girdle band. Okay, now this upper top is called hypovall, epiwall, and lower top is called hypovall, right? So this is the structure of diatom, and this complete shell, this complete body shell, this complete covering which is made up of silica is called frustules. Right. So when diatom die, frustules do not degrade. So we extract these frustule and use for the various purposes. Clear? Diatom clear? Clear to everyone?
Yes,
Sir, could you please explain this one?
Which one? The structure of diatom?
Yes, sir.
Okay, fine. Look up here. So the shell of diatom, look. This is shell of diatom. Look like a tiffin box. Like suppose that like this one. So this is our tiffin box. Suppose that this is a tiffin box. So diatom shell look like this one. So suppose that the tiffin box have two parts, the lower base part and upper lid part. So here the lid part shell we call it epitheca. This one, epitheca. So epitheca is upper part, and the lower part is called hypotheca. So it have two parts, epitheca upper part and hypotheca lower part. Fine. Again, this tiffin box have certain kind of design element over there, like girdle like design. So these are called girdle band. Fine. Now the upper surface of tiffin box is called epiwall, this one, and lower surface is called hypovall. And this complete external structure or external covering is called frustule. Now it's clear?
Yes, sir.
Okay. So please note it down. Chrysophytes. They all are aquatic. And some of them, they all are aquatic, and some of them are found moist. Some of them are found moist terrestrial habitat. So most of them are aquatic, but some of them are found in moist terrestrial habitat. Next point. They are free floating structure. Full stop. after the death of organism, they pile up at the bottom of after the death of organism, they pile up at the bottom of water body, and this and this thick layer of this thick layer of dead bodies called diatomoid. This thick layer of dead body called diatomoid or diatomaceous earth. Diatomoid or diatomaceous earth. Next point. The body is covered by siliceous. The body is covered by siliceous, means made up of silica. The body is covered by siliceous shell, which is transparent, and it is also known as frustule, and it is also known as frustule. Next point. They have chlorophyll A and chlorophyll C. This have chlorophyll A and chlorophyll C. Next point. Diatoms contain fucoxanthin. Diatom contain fucoxanthin pigment. Diatom contain fucoxanthin pigment, which provide them brownish color. Next point. Reserve food is oil and carbohydrate. Reserve food is oil and carbohydrate. Very, very important question. It is question of your medical examination. So make a star and note it down. About half of all the organic matter synthesized inward. About half of organic matter synthesized inward are believed to be produced by these diatoms. Clear? Is it clear to you?
Yes, sir.
Yes, sir.
Okay. Next, one more important thing, their economic importance, whatever I have explained you while I was telling this diatomite earth, like toothpaste making, sugar syrup making. So note it down, the economic importance of this diatomite earth. Note it down. Diatomite is used as cleaning agent. Diatomite is used as cleaning agent in toothpaste and polish. Toothpaste and polish. Even like you use certain kind of those hair gel, or what we call them. They are something oil-like substances, and like females pour them in their hair to make it more slippery, right? So they make their hair slippery, silky kind of hair. So these like those kind of syrup, or this hair syrup, we call them hair syrup. So these are made up of this one, diatoms and diatomite. Next, diatoms are also used in making filter for diatoms are also used in making filter for sugar syrup, oil, sugar syrup, oil, and antibiotic. So this is all about the diatoms, or we can say chrysophytes. So now come to the third subtopic, and that third subcategory for the photosynthetic protists, it was euglenoid. And the representative organism of euglenoid is euglena. Euglena is peculiar itself in this world, so you won't found any other piece like euglena. It is single piece, like it is single piece organism across the world, which have this kind of characteristic. So the characteristic feature of euglena is that it's not neither completely animal nor completely plant, right? Why? Because in presence of light, so suppose that light is present. In presence of light, what euglena actually do, it perform photosynthesis and make food. So it make food by the process of photosynthesis. So we call this kind of nutrition is autotroph. And in absence of light, so what they do in absence of light, they become consumer, they become heterotroph, they become consumer, they start eating organic particle, they become consumer, and then we call heterotroph. So they are the only organism on the world which have both kind of nutrition. So what kind of nutrition? Suppose that you get a question that what kind of nutrition is found in euglena. So euglena have the nutrition, mode of nutrition is called mixotroph. So euglena are mixotroph. They have mixed type of nutrition. They are autotroph as well as they are heterotroph. And the second important thing, euglena is actually animal cell. Why animal cell? Because it do not have any cell wall, and outer layer is that stretchable. It is made up of stretchable protein, right? So their cell structure resemble with the animal cell, and actually it bear chloroplast. So we can say this is the only animal cell which bear chloroplast or perform photosynthesis. So what is the status of euglena? Actually, euglena is called connecting link. Euglena is called connecting link between plant and animal. Plant and animal. So we call euglena is connecting link between plant and animal. Clear? Please note it down. Euglena is called connecting link between plant and animal. Euglena is called connecting link between plant and animal because it have characteristic of both because it have characteristic of both plant cell as well as plant cell as well as plant cell as well as animal cell. Right? So let me show you diagram of Euglena. Look, this is Euglena. One more important I would like to tell you, Euglenoid have generally two flagella, but Euglena have externally only one flagella. So internally these two flagella arise, but after some like after some distance or after some length, this smaller flagella it fuses with the longer one and they make single flagella. So normally Euglenoid means this class, the organism of this class have two flagella. Don't get confused, because sometime you will study that they have two flagella. So and you will say, oh, you have told me that sir, you have told me that Euglena have one flagella. So try to understand. Euglena have single flagella externally. At the point of origin, it have two flagella, right? In fact, every euglenoid, like all those organisms, including Euglena, which comes under this class, they have two flagella, one longer and one smaller one. But externally, Euglena have one flagella. Next. Another important thing is that flagella is for locomotion. They have stigma or eye spot. The stigma or eye spot are a structure which provide them sensitivity of light. So what is the utility of these structure? Look, whenever light is at the external atmosphere, what will happen? These Euglena, they will come out of from surface and they will start floating on the surface of water. So they come out of bottom, like the surface of bottom, they arise from there and they reaches at the top of the water surface, and to attain maximum amount of light and perform maximum photosynthesis. So whenever light is not there, so what happened? This stigma, they will receive the sensitivity of light from the stigma or eye spot. So and they will go deep inside the lake or ocean. Again, they have contractile vacuole. So contractile vacuole is not a structure which is found in a plant cell. Actually, this is the structure of animal cell. Contractile vacuole is used for removal of wastewater. Golgi apparatus is endoplasmic reticulum. These are normal cell. They have chloroplast for photosynthesis, mitochondria for energy generation. They have nucleus. Okay, fine. So this is the structure of Euglena. Is it clear to everyone? Clear?
Yes, sir.
Okay. So note it down, the characteristic of euglenoid. Euglenoid include, take the screenshot and then I'll remove this structure. Look. Note it down. Euglenoid contain Euglena-like unicellular form. Euglenoid contain.
Due to some network error, I left.
Okay, so what you missed? Did you miss anything?
No, sir, just 5 seconds before.
Okay, so no issue. So please note it down, the characteristic of euglenoid. First characteristic. Euglenoid include Euglena-like organism. Euglenoid include Euglena-like organism. Full stop. Next point. They have both autotrophic and heterotrophic mode of nutrition. They have both autotrophic and heterotrophic mode of nutrition. Full stop. Such mode of nutrition is called mixotrophic. Such mode of nutrition is called mixotrophic nutrition. Next point. Their body is covered by a thin and flexible structure. Their body is covered by a thin and flexible structure, which is called pellicle. Comma. It is made up of a flexible protein. Full stop. Next point. They have eye spot or stigma. Which work as photoreceptor. Next point. They have contractile vacuole. Which is a excretory organ. Help in removal of wastewater. Next point. Food reserve is carbohydrate. One last point. Reproduction takes place by binary fission. So that's it for Eulinoid. Is it clear to you?
Sir,
Yes.
sir, the function of eye spot is only to sense the light.
Exactly. You know, like you have a rain sensing wiper in your car. Like you have those doors which open when you reach over there, and they recognize something, the impact of light, and they open. So these kind of photosensor, you have photosensor. So that kind of, this eye spot is like that kind of photosensor. Clear?
Okay, sir.
And they have chloroplast, that I have told you, diagram only. So chloroplast is meant for photosynthesis, and this is the only animal cell in the world which can perform photosynthesis, or we can say this is the only animal cell which is autotroph. Note it down. One last point, one more point. They have chlorophyll A and chlorophyll B. Actually, chlorophyll B is characteristic of higher plant. They have both chlorophyll A and chlorophyll B. So each and everything clear about euglenoid?
Yes, sir.
Ifra, clear? Okay,
Yes, sir.
great. Now we have to start second class of Protista. So first class of Protista was photosynthetic Protestant. We did all Dinoflagellate, Chrysophytes, and Euglenoid, right? So in Dinoflagellate, Gonavalax, Gymnodium. In Chrysophytes, we did Diatoms and Desmids. Euglenoid, we did Euglena. Now let's start with the consumer decomposer. And in consumer decomposer, you will study slime mold. So slime mold, they basically resemble with the fungal, and some of the characteristic are there resembles with the animals. So sometimes they are known as fungal animal. We term them fungal animal. So second class is consumer decomposer protist. Then consumer decomposer protist will study slime mold. So slime mold actually basically they are saprophyte. So they are saprophyte. So generally they are saprophyte means they are depend for nutrition on dead and decaying organism. Decaying. So dead and decaying organism or organic matter. Organic matter. Maybe a plant part or maybe animal part. So whenever these material are not available over there, they behave like amoeba, and they behave like consumer, and the nutrition is phagocytic. Phagocytic nutrition. So in phagocytic nutrition, they eat bacteria and some other organism or organic matter. So they have both kind of nutrition. Fungal nutrition, so saprophytic mode of nutrition is fungi, characteristic of fungi. Phagocytic nutrition is characteristic of animal. So that's why they are also known as fungal animal. We call them what? Fungal animal. So they are also known as fungal animal, right? Look, so let me show you diagram of this one. Slime mold. So slime mold's diagram is very simple. So you know, this is diagram of slime mold. They have ectoplasm, outer protoplasm, endoplasm, like inner protoplasm, and there are number of the nuclei. So generally they are multinucleated. So they have multiple nuclei. So this is the structure of slime mold. Actually, they look like this one. They look like this one. So it's a structure of slime mold. So take the screenshot of. Yes. Yes, Vaishno. You want to say something?
No, sir. Sorry, sir.
Okay, no issue. Look, so this is the structure of slime mold. Take a screenshot of structure of slime mold, and let me tell you some more characteristic of slime mold. Take the screenshot. Look, so slime mold. I will show you one mold diagram actually. Actually, what slime mold do? This is the, you need not to cram it actually. What I am showing you, but I want to make you aware with their characteristic in depth. So slime mold, they are this kind of organism. The seventh one, solitary cell. It's a single cell of slime mold. So what happens at the time of scarcity of food? this is the feeding stage. At this stage, they keep on eating bacteria, they keep on eating other organisms. So what happened at the time of scarcity of food, they aggregate amoeboids. So these number of the amoeboid cell of slime mold, they get collected at one part and they pile up at one particular place. And after they make a colony-like structure. They again consolidate themselves and make a colony-like structure. After the colony-like structure, they make this kind of fruiting bodies, and these fruiting body develop spores. These spores spread here and there, and they are hard structure. On the favorable condition, these spore rupture and they again make amoeboid. So this is the asexual reproduction of slime mold. Now come to the sexual reproduction. What happens? Some of the two slime mold cell, they fuses, and the fusion of slime mold cell is called syngamy. Means two slime mold, they come closer and they fuses with each other. They are protoplasm fuses, then they are nucleus fuses, and they make zygote. After zygote, they divide by meiosis and make number of amoeboid cell. So this is the sexual reproduction, and this is asexual reproduction of slime mold. Clear?
Yes, sir.
So if you want, you can take a screenshot of life cycle of slime mold as well. Please note it down the characteristic of slime mold. Take a screenshot.
Yes, sir. Okay.
So we have written this characteristic of slime mold, and we have also written that they are called fungal animal. Right. Next point. They do not have chlorophyll. They do not have chlorophyll. And cell wall is not present, and cell wall is not present around. Cell wall is not present around them. Cell wall is not present around them. It is only surrounded by, it is only surrounded by plasma membrane. Full stop. Next point. They look like amoeboid cell. They look like amoeboid cell, and usually they have saprophytic mode of nutrition. Usually they have saprophytic mode of nutrition. Full stop. Sometime they become phagocytic. Sometime they become phagocytic and feed on bacteria or some other organic material. Next point. Reproduction is both sexual as well as asexual. Reproduction is both sexual as well as asexual. Next point. Slime mold resemble with both protozoa and true fungi. Slime mold resemble with both protozoa and true fungi. Full stop. This was all about the consumer decomposer Protestants. One more important thing, this is a question. Give me 1 second. Spore. Just a minute. Give me 1 second. There is a question of medical examination. It has been asked in AIMS examination, and I want you to jot it down. So, where is cellulose-like structure? They have some cellulose-like structure. Leave it. Actually, the important thing is that some of their stages they have cellulose-like structure, and this is a question. I was searching it, but I'm not getting it at this time. Leave it. I'll tell you next class, because right now I'm not getting exactly the question. I want you to write it down. So this was all about the saprophytic, like consumer decomposer, regarding the slime mold. And next, what we are going to study, note it down. We have time. So next class is protozoa. So the third kind of Protestant are protozoan Protestant. So in protozoan Protestant, actually they are the unicellular forms which have been taken out from Animalia kingdom. So they have been kept in this sub class that is called protozoan Protestant. So note it down. Protozoan Protestant. Very important. Already asked question of medical. Protozoans were studied by first Leeuwenhoek. So protozoan were first studied by Leeuwenhoek. They can be divided into further subgroup. They can be divided into further subgroup on the basis of their locomotory organ. On the basis of their locomotory organ. So first one is flagellated protozoan. Flagellated protozoan. So what is the example of flagellated protozoan? So let me tell you the example of flagellated protozoan. There is a flagellated protozoa, and the name of that protozoa is called Leishmania donovani. No, trypanosoma. Write the example. I will also tell you Leishmania. What is Leishmania? Trypanosoma. There is Trypanosoma gambians. Actually, Trypanosoma gambians is found in African country, and this Trypanosoma gambians is a type of protozoa, and it is responsible for a disease called sleeping sickness. It's a parasite and causes disease, and the name of disease is sleeping sickness. sleeping sickness. In this disease, host, or we can say patient, goes into deep sleep, like in coma like state, right? And you know the where this what is the reservoir of this this parasite? So Trypanosoma gambians, there is a reservoir. So reservoir is antelope. So antelope is an animal. In the body of antelope, this parasite is found, but they do not drop an impact. They do not affect antelope. So there is a fly, and name of that fly is CC fly. You know this CC fly. This CC fly, and we write what? CC, TC, TC, CC. So the CC fly first bite to antelope, and it take this Trypanosoma gambians. Again, it bite to human, and it inject this Trypanosoma gambians in human. And when this Trypanosoma goes inside the body of human, it causes sleeping sickness. And even due to this disease, person it damages, this disease damage the brain, and even most of the time person die. Clear?
Yes, sir.
Yes, sir.
Clear to everyone? Now the second example. So this was the first example. Second example is Leishmania donnamoni. Leishmania, both are very important for perspective of your NEET examination. So always take care of reservoir, take care of vector. This is CC flies vector and disease caused by this pathogen. So Leishmania donnamoni. Leishmania donnamoni, it causes kala azar. Kala azar. Actually, body become black. So that's what called kala azar. It's a fever. Kala azar or dum-dum fever. Dum-dum fever. It is also caused in South Africa or African continent. So Leishmania donnamoni is a parasite. It causes a disease called kala azar or dum-dum. So kala azar or dum-dum, it is spread by, means the vector is, it is spread by a vector, and that is also fly. Here it was CC fly, here it's a sand fly. So sand fly is a fly which spread this pathogen, and it causes dum-dum fever. So these are the two examples. In next class we'll study from Protozoa and Protestant. So in next class I'll tell you the structure of flagellated protozoa, how the protozoa work. And after completing this protozoa, we'll do virus. So did we taken care of virus? Have we covered the viruses? We haven't.
No, sir. We
So
have.
next
No,
class,
sir.
okay. In next class we'll discuss protozoa, and then we'll discuss viruses, and then we'll go ahead for next chapter. Clear?
Yes, sir.
Okay. So see you in next class.
Excuse me, sir.
Yes.
So what is the name of the vector in Kalazar?
Colors are sandfly.
Okay, sir.
Sandfly. This one. This is simple sandfly. It's vector. Clear to everyone? Okay. So bye bye. Take care. See you in next class.